Hello. Uh, Good afternoon, everyone. Thank you very much for joining us. Can you all hear me? Thank you. So, thank you very much for joining us. It is exactly 12:02 in my laptop. We will wait 3 more minutes. In the meantime, like Harrison can set up his presentation, and Harrison is here with us. And I'm just not going to go into too much detail. I because we know that he has a lot to discuss and share with you all, and want to give him maximum time. So once again, thank you very much for being here. And Harrison, over to you. You can share your screen, and it's the floor is all yours. Thank you.
All right. So thank you, Shazia, for that introduction. And can you guys all see my screen?
Oh,
All right.
Yes.
Thank you. So good afternoon, everyone. And as Dr. Shazia Hakim said, that my name is Harrison Cayatineto. And today I'm going to be presenting my final defense for my MS project. And today I'm going to be presenting my thesis project on potential microRNA-based inhibitor against the nucleocapsid protein in bovine viral diarrhea virus, a surrogate model for hepatitis C virus. So before I get into my presentation, I just want to share all of the approval letters that I had obtained in my first year of my MS program. And my thesis was approved by the Diné College IRB, the Legislation of Local Government, the Western Navajo Agency Council, and we also got the Nethatali Association approval letter, the Class C permit, and my master's thesis was also approved by the Navajo Nation Human Research Review Board. And here is the agenda for my master's defense with all the chapters with the acknowledgements and dedications at the end. So in my first chapter, the introduction, I'm only going to be giving an overall view of my chapter 1. But if you are very interested in learning more, I highly recommend that you check out our publication in the Journal of Human Virology and Retrovirology, because there's a lot of information in there that I can't really share today due to time limitation. So known as inflammation of the liver, hepatitis is usually caused by a variety of viruses called hepatitis viruses. And one of those viruses is hepatitis C virus, also known as HCV. And this virus attacks the liver, attacks the liver cells known as hepatocytes, causing inflammation. And HCV is actually part of a group of viruses called flaviviruses. And these are groups of envelope and RNA viruses that are mainly transmitted through mosquito bites and tick bites. While HCV, however, is mainly transmitted through contaminated blood, other blood products, and bodily fluids. And death rates and infection ratios due to HCV is actually 2 times higher among the indigenous population than the non-indigenous population in the United States. And most of those cases, the individuals are actually asymptomatic. So they are actually unaware of, their infection stages until they reach the last stage where recovery is almost impossible. While there are treatments available for HCV, but the effectiveness of treatment actually depends on the stage of infection and how early it is diagnosed. And even though there are treatments for HCV, there's actually no vaccine for HCV, which is why it continues to be a threat to global public health. So now I'm going to talk about a very tiny molecule that is naturally found in our bodies. And these are called microRNAs. And they're termed micro because they are extremely tiny to where they extend from 9 to 23 nucleotides long, while a mature RNA is almost 2,000 nucleotides long. So these microRNAs play a valuable role in our growth and development, our immunity, and they also play a role in something called the central dogma. And this is when our DNA gets turned into RNA, and then the RNA is then translated into proteins. But on their own, they are actually considered non-coding, and they have been used in antiviral treatments today. So this is the different stages of hepatitis. So during the early stages of infection, we start out with a healthy liver, but over time, it starts to progress into liver cirrhosis, hepatocellular carcinoma, which is another term for liver cancer, and then finally liver failure and death. And it's usually between these two stages is where the individual is finally aware of their diagnoses. So we wanted to find out the impacts of HCV on the Navajo Nation. But unfortunately, there really is no data on hepatitis and impacting the Navajo tribe. So instead of just focusing mainly on Navajos, we decided to go more broadly. So we decided to focus more on American Indians in general. So on the left are the cases due to HCV, and the one on the left are the death rates due to HCV. The x-axes are the cases per 100,000, and the x-axes are the ethnic groups. So as you can see with cases and death rates, American Indians had the highest cases of hepatitis and death rates as compared to the other non-Indigenous groups. So there are 7 different types of HCV known as genotypes. And there are 7 genotypes of HCV, with genotypes 1, 2, and 3 being the most common in the US. And not all genotypes are sensitive to treatment, and not all treatments are effective against all genotypes, especially genotype 3. So here we have a photo of the virus structure in the cartoon on the left. So this shows all the different structures of the virus particle. So we have glycoprotein, we have the envelope, then we have the virus's genome in the center, and then we have the capsid protein that protects the virus genome inside the virus particle. So in my thesis, this is the protein that I will be focusing on targeting. And the photo on the right is a real-life virus particle of HCV that went through a very special staining and viewed under an electron microscope. So what I learned about HCV is that it comes with some limitations and challenges. One is that it requires a biosafety level 3 lab, while the lab that I worked in the Hakim lab is a biosafety level 2, which is not suitable for handling this virus. And 2, it's that it's very difficult to grow because there is no ideal cell line to grow HCV in the lab. And also HCV requires a primary live host in order to grow. So it needs actually the liver cells in order to replicate. So I just thought, how can I find a treatment for HCV when I can't even work with it? And that was when I was introduced to this virus called bovine viral diarrhea virus, which I'm just going to only call BVDV today. So BVDV is currently still being used as a replacement model for HCV, studying its viral replication and also developing anti-therapeutics. So BVDV is actually the model system for understanding and studying the infection cycle of HCV. And again, here is a little cartoon structure of BVDV with the glycoproteins, the envelope, and the virus genome, and also the nucleocapsid, which is the focus of my thesis. And on the right is the actual virus particle that went on through a very simple, a very special staining and viewed under a microscope. So what I'm trying to do in my thesis is that I'm trying to find a new way to stop the HCV virus from attacking the liver by blocking its genome from making more copies. So basically trying to stop the virus's infection cycle. And BV/DV is used as a surrogate model for HCV because they are very similar to one another. And BV/DV is actually a close relative, but it doesn't infect humans, which is why it is used as a surrogate model when studying HCV. So to accomplish this, we use a lot of computer databases that already contain microRNA sequences that with the possibility to block virus replication. And we select those microRNAs that showed the most perfect similarities with the BV/DV virus's genome. So in my central hypotheses that we designed in my thesis is that if these microRNAs are considered non-coding, then we can use them to suppress virus growth by targeting a specific piece of the viral genome known as the mRNA. So if we find the right sequences that match with BV/DV, then we can hypothesize that those same microRNAs can be used or can align with HCV as well. And hypothetically, if functional proteins help with virus replication, then inhibiting them could more successfully inhibit virus growth. And functional proteins include like the glycoprotein, the envelope, and even the capsid. So some of the research questions that we are looking to answer in my thesis is that what microRNA sequences matched with the nucleocapsid gene of BV/DV, and I'm going to refer to this as NP, and the capsid protein, also known as CP, of HCV and its genotypes. Which microRNA showed the most promising in inhibiting effects against BV/DV? And which microRNA from question 1 not only align with BV/DV, but with HCV, or not just BV/DV and HCV, but with the rest of the flavivirus members, because they all come from the same family. So to do this, one of the aims is to use tools to run sequence alignment analysis using computer databases like MIRBase, MIRDB, and also the NCBI Basic Local Alignment Search Tools Method nucleotides. I know that was a mouthful, yikes. But today I'm just going to call it BLAST-N. And then also also utilizing A2, which is all of the laboratory work to actually insert these microRNAs and observe them for any or confirm any suppression of our virus, the BV/DV virus. So kind of think of it like a puzzle that is complete, but is incomplete due to a mismatch. So in the figure on the left is the microRNA that's represented in blue, and the virus genome is represented in yellow. So you can see that this microRNA matches and sticks with the genome because of similarities. And the image on the right, the green puzzle pieces represent the BV/DV genome, and our microRNA is represented by this mismatch piece. And you can see that it's not the same piece as the rest, but it fits perfectly. So our microRNA will stick to our BV/DV virus like this wrong puzzle piece, which then in the process, not allowing our virus to grow and infect our cells. So now we're going to go on to chapter 2, which is where I collected all of the microRNAs by using a lot of computer databases. So this is just a quick flow chart of how I did my sequence comparisons. So in steps 1, 2, and 3, I had to actually first collect all of my virus's genome and then match it with the microRNAs. So when we find the right microRNAs, we should see something like this that we see in this zoom figure, to where this microRNA matches with the virus genome based on similar sequences or similar sequence comparisons. So if we find the right microRNA that shows this same matching pattern, then it could actually lead into step 5, where the virus's mRNA is degraded and destroyed and no virus is made. So the first thing, again, I had to do was that I had to collect all of my target virus's genome sequences. So in the table here, we have the virus name, we have the accession number, the gene name, and also the sequences responsible for this gene, which is the nucleocapsid and the capsid gene of my viruses. So it was these sequences that I matched the microRNAs to. So this is a screenshot of the BLAST home webpage. So in order to run these sequence comparisons, I entered my microRNA sequence in the query sequence box, and I entered in my virus's genome sequence into the subject, and then hitting BLAST to run the comparison. So in the results here, I just want to turn your attention to the yellow box where it says no significant similarities found. So basically means no alignment. But that's okay, we can try again. So just imagine that I went back to that homepage where we see those two boxes. A different microRNA is entered in the query box, and the virus sequence is entered into the subject, and then hitting BLAST again. And then we get a result page like this. So if you look at that, we got an alignment. Hooray! This means that there is a similarity between the microRNA sequence and my virus's mRNA sequence, with the query representing the microRNA and the subject representing our virus. But there are other information here that I won't really get into much detail because of time. So I just so these numbers here just tell us how good our alignment is. So usually I don't always get a single alignment like you see here. I also get 5 to even 8 alignments. So meaning that one microRNA aligns at 8 or 5 different locations on our virus's genome. So all the data that you see here were recorded and inserted into an Excel spreadsheet with match indicating in green, while red indicates no match. And I spent over there every day collecting alignment data. I went through all the microRNAs, aligned them with the BVDV virus and the other HCV virus genotypes, and used them for further analysis. So with these two databases, MirBase and MIRDB, I analyzed over 5,200 human microRNAs, aligned them with the BVDV virus's genome, and with the HCV virus and its genotypes. And then, like I said, I wanted to go even further to where we find these tiny microRNAs that not only match with BVDV and HCV, but it matched with all of the other flavivirus members because they all come from the same family. So altogether, 5,200 microRNAs times 13 virus genomes equals 67,600 alignments I performed. Whew. So here's another Excel spreadsheet of all my alignment data with the name of the microRNA, the accession number, identity, score, expected, etc. So again, match is indicated in green, while no match are indicated in red. So when I first started doing all of these alignments, I matched all the microRNAs with BVDV first, and I only selected those that only showed a match with the microRNAs. So that's why here in the red box, it says match all the way down. So, so after I ran all my alignments, I looked for those one microRNAs that match across this group of viruses. So meaning, for example, here, there's a microRNA that had a match with BVDV, and then it matches with HCV1, HCV2, HCV3, 4, 5, and 6. So this pattern was what I looked for throughout my whole Excel spreadsheet. So as you can see in this example, there's one there and there's one here. So again, I went through my entire Excel spreadsheet and looked for what I'm calling today ideal alignments, and I found 180. So this is just a little a graph that shows how many microRNAs I started with and how many microRNAs I started now, or I ended it with now, excuse me. So in the beginning, I started with 1,200 microRNAs. Then I looked for those ideal alignments that I just talked about in the previous slide, and I ended it with 180. And because of time, we couldn't go through all 180. So we excluded the microRNAs that had more than 2 alignments and kept the ones that had less than 2. And in the end, we ended up with 9 microRNAs. But then we found something exciting, that we were actually able to find one microRNA that matched with all of the flaviviruses as a whole. And we were pretty excited about this. So here are the 9 microRNAs that were selected as possible candidates. So I'm not going to go into all of this information, but I just wanted to show you the name and the name of the microRNAs that you see here. And we use these for further analysis. So these microRNAs, I aligned them with BVDV, and the results here in this box, I don't really want to go again because of time. I don't want to go into all of this details, but I just mainly wanted to focus on the location on subject, which means this is where our microRNAs, where they aligned at on our virus's genome. So in order to compare them, I used a dot plot and plotted the start of the alignment and the end of the alignment. And this is what we got. So the graph on the left shows the start of the alignment and the end of the alignment is on the right. The y-axis is the alignment position, and the x-axis are the microRNAs. So as you can see that the dot plots are all scattered and they don't show any similarities. But that's okay, because BVDV again is just our test virus. So I did the exact same thing with the HCV virus and its genotypes. So I plotted the start point and end points, and they came out like this. So again, the left is the start of the alignment. The one on the right is the end of the alignment. And this is for HCV1, HCV2, 3, and 4. So as you can see, just by looking at them, they all look fairly similar to one another, meaning that these microRNAs are potentially matching at the exact same place on our virus's genome. And here we have HCV5 and HCV6. And again, the start of the alignment is on the left and the end is on the right. So the x-axes are the alignment positions, and the x-axes are the microRNAs. And just like how we saw with HCV 1, 2, 3, and 4, we see a very similar pattern here too on HCV 5 and 6, meaning that the microRNAs are potentially aligning at the exact same place on HCV 5 and 6, as well as on the other HCV genotypes. So now I'm going to talk about another microRNA called 65105P. So this microRNA actually aligned with HCV genotype 1. And again, I wanted to align this microRNA sequence with not only BVDV, but with the other genotypes of HCV. And this is what they look like. So in the box here, we have the virus here. We have the BVDV that was compared with this microRNA, and we have the other HCV genotypes here. And just like how I did where I used a dot plot to plot the start of the alignment and the end of the alignment, this is what it looks like. So even with, so let me start over. So again, again, on the left is the start of the alignment, and the end and the end of the alignment is on the right. The x-axis is the alignment location, and the x-axis is the virus capsid genome. So you can see, even though there are some slightly differences, you can see that they all eventually match at the exact same place. So meaning that this microRNA also is aligning at the exact same place as all of the other microRNAs. So now another microRNA that I want to talk about is 548D3P. But like I said, I'm only going to give a quick summary. But if you are highly interested, we recommend you that you check out our publication on Frontiers in Bioinformatics. So here is just some more details about this microRNA. And I'm not going to get into all of this, and we'll just move on to what the significance of this microRNA is. So here in figure A on the left, the x-axes are the number of alignment positions, And the x-axis are all of the flavivirus's capsid genome. So you can see that it has the highest number of alignment on yellow fever. The second is Zika. The third is West Nile, and the other one also on BVDV. And the figure on the right just basically shows the highest number of alignment. So the highest number is represented in red. So the highest number is on yellow fever virus. So everything that I talked about now was all of the biology, the bioinformatics part. So basically all the computer work. So now we're going to actually go into the laboratory testing to where we put these microRNAs to the test. So this is just a little overview of my methods. So in chapter 2, I already talked about how I selected my microRNAs. So now we're going to go on into the in vitro lab assays. So after we've selected the 9 microRNAs, we actually had to purchase them from a facility called IDT. So once we got those microRNAs, we prepared them for the experiment. And then we used MDBK, or we grew MDBK in Hep G cell lines until they reached the right growth ratio. And once they reached the right growth ratio, we had to use my virus stock solution that you see here. And this is highly concentrated with my virus. So it means that there's high numbers of virus in this tube. So we wanted to actually bring this high concentration from high to low so that we can use them for something called a plaque assay. So if you look at this little figure here, you can see that this is called a plaque assay. So the purple background is a dye called crystal violet, but we can see these little clear spots, these little white spots. This is where our virus is infecting our target cells, our cell lines. So if our microRNAs work, we should see less of these little clear spots and see more of the purple. That is if they are working. So this is theoretically what we're guessing that is what's happening in our cells. So here we have the cell membrane, which is our liver cell. Then our virus infects the cell, releasing its virus genome, which is shown in yellow, but at the same time, injecting our microRNA. And then once the microRNA recognizes the same sequence that it has on our virus's genome, it attaches, then virus translation begins. Or should I say virus growth begins. But once it reaches where that microRNA has attached to, it stops, meaning that another virus will not be able to grow because it was stopped by this microRNA. So here is actually one of my 6-well plates from one of my experiments in the lab. So here you can see in the background that it's all purple, but you see little black dots. These little black dots are where I marked where the plaque assays are. So as you can see here, there's a mark here, there's a plaque there, there's another mark here, 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 and there's plaques there, there, and there. And here too, there's plaques here, 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 there, there, there. So I went through all of my 6-well plates and looked for these plaques and graphed them in a growth curve analysis. So in order to get an idea of what a growth curve looks like, it will look something like this. So here is a growth curve of the virus when it infects our cell lines. The y-axis is virus growth, or how much virus is infecting our cell lines. And the x-axis is the serial dilution. So you can see that a growth curve makes this really wide S, indicating that the virus is indeed growing in our cell lines. But if we add our microRNAs, it should look something like this. So if our microRNAs work, if you see in the red line, this represents our cell lines infected with the virus, and we're treated with our microRNAs. So if our microRNAs work, our growth curve should actually decrease, indicating that the virus particles are actually getting lesser. That is if our microRNAs work. So now that we see what a growth curve looks like, let's see what they will look like in real life. Ta-da! So on the left are the MDBK cell lines, and the one on the right are the Hep G2 cell lines. The y-axis is the virus titers, so basically how much virus is infecting our cells. The x-axis is the seral dilution. Our control is actually just the MDBK infected with BVDV, but the other colorful lines indicate treatment with our 9 microRNAs. So as you can see in both MDBK and Hep G2, the growth curve with the microRNA treatments are actually much lower than our control, indicating that maybe our microRNAs are indeed blocking our virus from growing. So now that we see that how our microRNAs are playing out, we then for the top 3 the most inhibitory effects from both MDBK and Hep G2. So in MDBK, these are the top 3 microRNAs that showed the most promising inhibiting effects. So we found that microRNA 6851, 3064, and 574 showed the most inhibitory effects. So here in the graph, you can see that the y-axis is the virus titers, and the x-axis is the seral dilution. Our control is the black line, and you can see that the growth curve with the microRNAs is actually a lot, has decreased a lot more than compared to our control line. And the figure on the right is just basically the total virus titers counted. So basically all of the points added up and compared them to the microRNA treatment side by side. Black indicates the controls and the gray represents the microRNA treatment. So you can see that the virus growths are actually much more lesser than our control. And this is with Hep G2. And again, we can see that the lines that were treated with the microRNAs are actually lower. So we found that microRNA 1180, 876, and 3064 showed the most inhibitory effects. And here again is the total virus titers count. So as you can see again, that black represents our control, and our microRNA treatments are indicated in the other gray, I'll just call them gray bars for now, indicating that these microRNAs are in fact inhibiting and blocking our virus from growing. So then we have 6510. So with MDBK and Hep G2, we see that the line with the microRNA treatment is actually a lot lower than our control. So indicating that this microRNA is also preventing our virus from infecting our cells. So again, this is just total virus titers count. Again, the black indicate our control and the gray indicates for the microRNA treatment. And as you can see that the bar with the microRNA is actually a lot lower than our control bar. So right now I just want to visit my methods one more time because this part of my laboratory assay, I mainly focused on seeing how the microRNAs play out with our virus growth. So that's why we looked at a lot of growth curves. But now I want to see how they play out transcriptionally. So basically saying, are my microRNAs really binding to my virus's genome? So to do that, I had to go through RNA extraction and then go through a really cool process called reverse transcription PCR, which is where we actually take our RNA and turn it back into DNA. So after we have successfully amplified our DNA, then we would look at the bands under gel electrophoresis, which just sounds like a really long word, but it would look something like this. So here is our gels. This longer pattern of white lines indicate our DNA ladder, which tells us how big or how small our DNA bands are in size. And you can see these little white lines, these are our DNA bands. So the one on the left is not treatment with the microRNAs, and the one on the left were treated with microRNAs. So if our microRNAs work, then we should be seeing significantly skinnier bands, maybe some fainter bands, or even no bands at all. But if our microRNA treatments don't work, we should see much thicker and brighter bands like you see in this one on the left. So now let's look at my real gel results. So here the DNA bands are indicated by this arrow. So these are all the DNA bands that you see on top. So the first two rows are the cells infected with EVDV but treated with microRNA 574. This pair were treated with 3064. This pair were treated with 6851. And these last two are our controls that went through no treatment. But as you can see with microRNA 574, the bands do appear much more fainter than compared to our control. And on and for 3064, we see that there are some bright bands here, but they are actually relatively smaller and skinnier than compared to our control group. However, with microRNA 6851, we still see some bands here, which indicates that maybe the microRNA didn't bind, or it could significate that the PCR didn't amplify where the microRNA has attached to. So this is with MDBK. So now let's look at the gels that were performed on Hep G2. So this is with Hep G2. So again, the first two were the Hep G2 cell lines infected with BVDV, but treated with microRNA 876. These ones were treated with 1180, and these were treated with 3068. And the last two are our controls that have no microRNA treatments. So as you can see by looking at our DNA bands, we see that the bands are actually significantly fainter than our control. So this actually indicates that I may have successfully prevented my virus from growing by having the microRNAs attached to our virus's genome. So here again is another gel results, and this is with microRNA 6510. So here on the left is the y-axis is the percentage of virus mRNA expressed. So that just basically means how bright the DNA bands are. And the x-axis are our samples. So our controls are indicated in black. And the 6510 microRNA treatment are indicated in these gray bars. So as you can see that as compared to our controls, they are actually much smaller and much more lesser. And looking at our gels, we can see that. So now let's look at the gels. So again, the microRNA here was treated with 6510 on MDBK, and this one was treated with Hep G2. And I'm sorry for the noise in the background. I'm sorry. And the control are just the cell lines and virus with no microRNA treatment. So as you can see that the bands with the microRNA treatment is actually fainter than our control bands, significantly saying that my microRNA did indeed bind to our virus's genome, leading to inhibition of our virus. So now we're going on to chapter 4, which is the interview with Navajo elders. So in ethics statements, my method was actually approved by the Diné, or by the DC IRB, the chapter house, and then it was also approved by the Western Agency, and it was also approved by the Navajo Nation Hatahtli Association, as well as the Navajo Nation Review Board. And again, this study was actually voluntarily, so the subject had the choice of declining the interview or accepting it. So here are the methods from my interview. So this was a face-to-face interview that required 30 minutes to an hour with some note-taking. And usually it took place in a conference room or classroom. And it was, and my interview involved asking questions that involved the knowledge regarding HCV, what Navajo language is said for HCV, and what are the traditional practices, and what other things that we scientists need to improve to prevent HCV. So I'm only going to give a quick introduction to my results because I have because of limited time. So only 2 participants were interested in the study. So in general knowledge, when they hear the word hepatitis, they think of disease and or sickness. And what I actually came to find out was that there was actually no knowledge of HCV impacting Navajo, the Navajo community. And usually the Navajo language for hepatitis usually translates to liver disease or liver sickness. And with diagnoses, there is no way to recognize or diagnose hepatitis without actually going to the hospital. So, and there are no traditional medicine used for HCV. And I also asked, what are some of the ways to prevent the transmission of HCV? And both participants actually said that education, spread awareness, and also testing for HCV at early stages could potentially help with preventing the transmission of HCV. So the first aim of this study was to actually find the right microRNA that showed similarities between with our virus's mRNA genome and test them for viral uh, inhibitory effects. And in the second aim was where we actually utilized the microRNAs and use them in the lab to see if they could actually work at preventing our virus from infecting our cell lines. And as Stan Vang J reported in 2012, who actually found that microRNAs, or mature microRNAs, showed the most promising inhibitory effects. And we also found the same thing, that these mature microRNAs that we discovered are more developed and bind more effectively than the immature microRNAs. And as Zhang X et al. Said in 2021, who said that structural proteins are the most targeted structures in therapeutics, and we totally agree because our study found that targeting the capsid protein actually led to more inhibitory effects of our BVDV virus. And one of the future aspects of the Hakim's lab is to actually use a micro injector, which we already have in the lab already, and actually inject the microRNA 548-3P and test it for any inhibitory effects, because the 9 microRNAs we got were the only ones that we were able to test, but we didn't receive this microRNA yet. So this is one of the future aspects of Hakim's lab, is to test this microRNA next. So through this study, it was actually able to predict a few microRNAs that showed similarities to the BVDV, HCV, and the flavivirus families. And out of 10 microRNAs, and a total of 10 microRNAs were identified out of over 67,000 alignments, and nine of these 10 were taken from MIRBase, and one was taken from MIRDB. And 548 not only showed alignment with BVDV and HCV, but again with the whole flavivirus family. And through our PCR and our jet electrophoresis, it did confirm the presence of the microRNA attaching to the BVDV virus in the host cells, which led to significantly fainter bands as compared to our controls that had much more brighter bands. So after completing a whole set of plaque assays, we found that these 9 microRNAs could potentially be used in antiviral therapeutics against HCV and its whole family. And also through this study, based on the interviews, that there still needs to be awareness spread about HCV impacting the Navajo people, because the cases are high among the indigenous population. So there has to be a spread of awareness and educate Native people about HCV. So again, there were limitations that I experienced, like back in the early stages of my presentation, that HCV was highly infectious and it required a BSL level 3, while the lab I worked in was a BSL level 2. And additionally, there is no cell line available for HCV. And that's why a surrogate model was used, because it was non-infectious to humans and it also hold a lot of structural similarities to HCV. and that is the BVDV virus. So overall, this study was mainly a bioinformatic-based in vitro study that provided the initial first-hand results for a potential therapeutic agent that needs further testing for in vivo experimentation. So basically using animal models to test these microRNAs. So there were a lot of references that I used in my research, but these are the top 6 that I chose as the most important. So what I learned throughout this journey of my MS journey program, so I could just say that I learned a lot. Learning the art of teamwork, building collaboration, and also learning about the ethics of research, both in the Western way and also in the Navajo way. Especially the Navajo ethic research systems, and I'm really proud that my master's thesis may have opened the doors for the next future batches of students who are ready to tackle complex research and make a difference in the world. So now we're getting into the acknowledgements. So first off, I would like to acknowledge Diné College for giving me and the students the opportunity to conduct research in our lab, in the labs. And also acknowledging our sponsors like NSF, NIH, and the Department of Defense. And who none other to start off my acknowledgements than with my graduate advisor and mentor, Professor Dr. Shazia Tabassum Hakim, who took me in as her student and actually showed me the importance of not giving up. And Shazia did not give up on me when I was at my lowest point. I always felt like I always crumbled up a paper, threw it up, but she picked it up, unraveled it, and helped me again. And so I just want to say thank you, Shazia, for helping me reach one step of my goal of becoming a virologist. I wouldn't have done it really without you. And also acknowledging my graduate committee members, Dr. Christina Me, who was the one that actually mentored me and helped me design my consent form and also my questionnaires. And also I'd like to thank Omar Bagasra, who actually for his expertise in working with microRNAs and providing guidance and support. And I also like to acknowledge Dr. Ojo from Tuba City, who always showed up to all of my talks and always showed his own support and guidance as well. And also to Diné College faculty like Selena, who helped with all of my travel protocols to Tuba City, and also Dawn Robinson and the Dean of STEM, Dr. James Tuck, for their continuous support. And I'm sorry, Dawn, this was the only picture I could find of you, so I just used it for now. But I just want to say to you all, thank you so much. And again, we're going to Tuba City. I have a lot of people to thank from Tuba City as well, like Jovita, the guy, the security who actually watched over me when I stayed in the lab at late at night to almost midnight. And sometimes she would always see me drink a cup of coffee and ask me, how many cups is that? And I would say 3. So thank you again, Jovita, for always watching over me in the lab when I had to stay late at night. And also Cody, the custodian, and also Allison Bigay, who always gave me access to the science lab and the research trailer when I needed to. And also thank you, Allison, for driving us to Flagstaff for the NAU conference. So, and also to my colleagues, first starting off with Ian Nez, who has kept my cells alive when I wasn't on campus, and also Vanessa and Joycelyn, who always cheered me on every time when I ran back and forth from the research trailer to the research to the science lab, and always reminding me to eat because I always skipped lunch, I always skipped breakfast and even dinner. And they even wrote on the whiteboard in the research trailer, Harrison, make sure you eat. And I just want to say thank you to you all. So now going back home to my coworkers at Navajo University, I really want to give a big thank you to Dr. Palmer Natongo and Irene Aniyangwe. Excuse me, sorry. Ooh, sorry about that. But again, I just want to say thank you to Dr. Palmer and Irene Aniyangwe for their support and allowing me to actually go to Tuba City and do my work and taking a week off from work. And I also want to say thank you to the students who also supported me, who gave me all of the love and comfort when I felt like I couldn't um, continue with my MS. And I can't remember if it was either Deborah or David who told me that when I didn't feel like I didn't have no one to do this for, at least do it for us, is what they told me. And I always held that close to me when I felt like giving. And also acknowledging my family. I do have a big family and I don't have all of their pictures with me, but I just want to say thank you to you all for always being there for me and always supporting me whenever I felt like I couldn't go on. And also thanking my furry friends like our cat Junior, Pixie, and also Nanook, who also were kind of like my my therapists who always comfort me and show me love also. So now finishing off with dedications, I just want to say thank you to my mom who has always been there for me since the day I could walk. And she is my auntie, really, but I grew up calling her mom because she took me in when I was so small, was young and tiny. And all of her love and her comfort and all of her soft hugs is what got me through my master's degree. And thank you, Mom. And I'm also dedicating it to the loving memory of my dad, who is actually sadly no longer with us here on earth and made his journey back home to his creator. And I can't and I always remember how happy he was when I told him that I was doing this master's degree. He was so happy for me that he kept telling me, stay in there, you can do it. And I did. I finished it and I owe it up to my dad. And I love you all, Mom and Dad. And with that, I'll just leave it for any questions. Thank you. And I'm
Thank you.
sorry for and I'm sorry for all the noise in the background. I wasn't expecting that, but thank you so much for coming.
Oh, hi, Harrison. I have questions.
Yes.
First of all, that was a really incredible presentation. I love the way you tied everything together from the bioinformatics to the experimental. I
Thank
did
you.
come in a few minutes late. So my question is about the selection of your microRNA. I noticed that you were searching a database of human microRNAs and utilizing one of those. Why? I mean, why were you searching human microRNAs rather than just, designing and targeting a piece of the whatever the viral gene encoding the capsid? It seems almost like, and I could be wrong, that could um, detract from the drug if you're putting a human molecule, injecting it into humans, could interfere potentially with expression of some of our human genes. So that's my first question.
So I don't really have an answer for the first part of your question, but I can say that the reason why we used human microRNAs was because that we're actually finding an antiviral drug for humans. So we kind of figured that using human microRNAs would be more suitable for developing a viral drug. But for your other question about if that could cause any side effects, and that's where that in vivo experiment comes in. So right now it just provides the initial results. So this could set the bar for doing more in vivo experimentation. So using like animals models. But like I said again, this is just the first initial step. So we haven't really gone towards the animal studying yet.
A wonderful job. I really loved your presentation. Have you thought about using, instead of quantifying the level of inhibition from the microRNA by using the faintness or the strength of the bands, using a more quantitative procedure like qPCR to quantify inhibition?
So because the virus that we were working with is an RNA virus, so we had to kind of turn the RNA back into DNA. So
Yeah.
once the RNA turns back into DNA, then it gets amplified again. So so that would be kind of the only method we could possibly do because money is really tight right now. So the only
Right.
thing we could do was the reverse transcription PCR.
Yeah,
And
that's a really good start, I think, though. That's,
yeah, thank you.
yeah, wonderful job. Thank you.
Thank you so much.
Any other question? Remarks?
Hey, Harrison, it's Vanessa. Good job. I really enjoyed your presentation, and I'm so happy to see where you've come, seeing you in the lab and cheering you on. And I'm just so happy to have you as our mentor too. So, that way we can ask you questions or anything that we need and sharing your knowledge. It's really, I really feel honored to be working beside you, and it was good job. And Shazia too, you are awesome. We love you. Always there for us. So I cried when you were doing your remarks. So I'm very sensitive. But good job, Harrison. I really enjoyed it.
Thank you so much.
Dawn? Any comments?
Hi, guys. Well, I'm beyond impressed, Harrison, as I've said to you. And this presentation was really good, too. You did a really good job, simplified a very complex area of research. And I'm going to have to study it, Harrison. You're forcing me to study like a student again. It's really good. I'm very proud of you. Yes, sir.
Thank you so much. That means a lot.
Well,
And I see
Thank you.
where acting president, Dr. Husky is also here. Dr. Husky, VP Husky. Not VP, sorry.
Good afternoon, everybody. I'm okay. My original position is VP, but I'm really happy to have joined this presentation. And Harrison, I know doing a graduate study is not easy. I think first and foremost, you need to give yourself a pat on the back for getting started with the program and actually finishing the program. And then just the content of your presentation speaks volume. I think that on the outside, there are times that the college gets the perception of it's an easy institution and, there's not a lot of challenging in learning. But when I sat here and witnessed your presentation, you learned a lot. And for someone who comes from a background of, occupational therapy and have gone to medical school, I'm so impressed. One, that you are Native American. Two, you're Diné. And three, you're doing your graduate studies from Diné College. That's just layers and layers of things that you can walk away at graduation knowing that you have paved the way for others that are coming behind you. And, this is a degree that nobody can ever take away from you. It's yours now. You've earned it. Granted that everybody votes that your presentation is accepted. It's yours, but what you do with it, again, is at your ownership. I can only ask that in time you give back to the people, because our people need it. And I'm getting up there in age, and one of these days you might be my medical doctor that needs to do some high level treatment on me. And I'm going to say, yes, I went to your graduate presentation. So there's so much I want to say, but I think all in all, you have a right to be happy and to be proud of yourself. And it's always great to hear a student acknowledging the people who are in their background that pushed them and helped them succeed. So don't ever forget your grounds, and don't ever forget your roots. And you know that just speaks volume on you. So I commend you to the fullest.
Thank you so much.
Thank you very much. Dean Tut,
Yes, thinking back about a year and a half ago, I think Harrison pretty well was in a state of distress and all of it. But it was good to see him to come through and show his knowledge about the microbiology side of it. And just thinking back, when we had COVID-19, it was basically a lot of virology being talked about it. And these are one of the areas that a lot of our students were going to be going into. So I see some of it, the work that's going to come forth from, our own students. And I'm very happy to see him pull through. So I'd like to thank Dr. Hakeem, graduate committee, and the committee members that guide Harrison. Thank you.
Thank you. Thank you so much.
thank you very much, sir. VP Lewis, would you like to have some comments?
It looks like there was a question asked by Tan Clyde. It says, where are the microRNA sequence coming from? So most of these microRNAs were already extracted from plants, even some humans, insects, and even other animals. And they were already extracted and entered into these databases. So that's where they mainly come from, mainly from the plants and insects and humans.
But
Yes. So I have a follow-up question to that is, if it was being applied, how would you use the plants or the insect sequences? How does that work?
So in the databases that I presented in the first part of my presentation, where the like MirBase and MIRDB, they have already that list of microRNAs from plants. So we just basically copy and paste the sequence and then match it with the BVDV virus's genome, and then just match them all down like that. Like how I said in that that BLAST webpage where we have the box where we enter the microRNA sequence, and then we have a second box at the bottom where we enter in the virus's genome, and then just hit BLAST and it just compares them. It just compares the two sequences together. So that's how I did it with these microRNAs, but it can also be done with the plants also.
Shazia, this is Bo.
thank you.
Can you hear me?
Yes.
Okay. Yeah, I just wanted to say you did an excellent job, Harrison. Proud of you. And I'm just wondering how we could set up the lab. How can we increase or improve the lab so you can do the actual work? That would be great too, so that you can really see the results. That would be nice. But just based on what we have right now, you did an excellent job and you made it work, and it could help some people. And for the next phase of this work, I think it can be helpful to our Native people, because they're the ones that are the highest in this diseased area. So anyway, that would be great. So very proud of you and thank you for taking the initiative to take on such a hard job and also making it understandable for us lay people too. So thank you.
Thank you so much. That means a lot. Thank you.
Mhm.
Okay, so thank you very much, everyone. And I think we do not have any more questions or comments. Again, it was lunchtime and most of you have your classes coming up. So please, thank you very much again. And we will bring forward some more presentation with our more students. And just thank you for all the supports, your presence, and everything. Good job, Harrison.
Thank you. Thank you so much, Shazia.
Thank you very much.
And Vanessa, Joycelyn, I will be cheering you guys on too and making sure you guys eat next.
Thank you.